Welcome back to the playlist on amino acid catabolism. In this video, what we're going to do is we're going to look at histidine catabolism. Now, this particular picture I got from this website, and by the way, this site up here that's on the top right of the screen, this is a really good website if you're interested in learning biological pathways. Very good. It has pretty much everything you'd ever want to know. Okay, so that's just a really good website. And I got this picture from there. Okay, and this picture is going to have more than we're actually going to deal with in the actual catabolic pathway. What I'm going to do is I'm going to focus on the reactions. I'm going to focus on the reactions that are shown in Leninger's Principles of Biochemistry, 6th edition. Okay, and what it says right here, histidine biosynthesis, note that the process of histidine biosynthesis does not occur in eukaryotes. Okay, it only occurs in bacteria. So this catabolic pathway occurs in both eukaryotes and bacteria, but the biosynthesis of histidine only occurs in prokaryotes. Okay, so having said that, let's now look at the catabolic pathway for histidine. And also, one thing to note is we actually have two mechanism videos on two of the enzymes. They're the first two enzymes. So the first enzyme that we have a mechanism on is histidine ammonia lyase, and that's going to be this enzyme right there. So histidine ammonia lyase. And the second enzyme that we have a mechanism on is this one, which is urocanate hydratase. Okay, so let's go over the first one, which is histidine ammonia lyase. And histidine ammonia lyase is basically going to remove the ammonia group. It's going to remove this group right here from histidine, and in the process, it's going to generate this alkene, right? And notice that the alkene is in the E configuration, right? Both high priority groups are on opposite sides of the double bond. And so you could say that the carboxyl group is going to be trans to the imidazole ring, and as a result it gets the E configuration. And certainly go watch the mechanism of that video. And that particular enzyme is going to require a special cofactor called a methylidine imidazolone. Methylidine imidazolone cofactor. And the reason I show you that mechanism is just because it's interesting. And for the most part, you won't really see that cofactor ever again. And actually, it's a post-translational modification of the enzyme. So it's a very strange cofactor, and it actually is a part of the enzyme. Post-translational modification of histidine ammonia lyase. And of course, that's going to remove ammonia, and you're going to generate this molecule right here, and this molecule is called urocanate. This is called, and you can see it right here, this is urocanate, okay? And urocanate is going to get hydrated in an extremely complicated mechanism. And the enzyme that does it is called urocanate hydratase. Now, what I want to also mention about urocanate hydratase, which is labeled as 2, is this particular enzyme is NAD dependent. It's an NAD-dependent enzyme, and there's no net NADH that's formed, okay? So, in other words, you're going to, in the mechanism, you're going to attach urocanate to NAD, then you're going to hydrate urocanate, and then NAD is going to be removed. So there's actually no NADH formed in the mechanism. NAD is just used to facilitate the hydration of urocanate, okay? And if you actually look at the mechanism, which I recommend that you do, NAD is still an electrophile, and excuse me, this particular mechanism is essentially going to be an addition reaction of an alkene. It's an addition reaction. And also notice one other thing that's interesting about it, okay? Urocanate has our characteristic aromatic imidazole ring, right? And even though we hydrate the imidazole ring, we still retain this aromaticity at the end of the mechanism, okay? And also note that this double bond right here has been totally reduced into an alkane. Excuse me. Okay, now here's what I want to point your attention to because it's really important. Okay, this structure that I have right here, okay, this structure is the enol version. It's the enol version of something called imidazolone propionate. Imidazolone propionate. And it's specifically, and keep in mind, it's the enol form. Now, the enol form is what's initially formed by the enzyme, okay? But what's going to happen is the enol imidazolone propionate is going to tautomerize. So if you notice, right, this particular group right here, here is your enol part of the molecule, right? And again, we're just looking at one resonance structure, right? We can draw multiple resonance structures. And in fact, the double bond character exists throughout the entire imidazole ring. But essentially what's going to happen is it's going to tautomerize. And so it's going to tautomerize into this guy right here, which is a carbonyl, right? And it turns out that the equilibrium actually favors the carbonyl. And that's strange, right? Because you say, okay, well, the imidazole ring, which is right here, right? This has an imidazole ring, whereas the other one, which I'll do in yellow, this one does not have an imidazole ring, right? If you notice in the second one, which has the carbonyl, the imidazole ring has been destroyed, right? It's no longer aromatic. And my question to you is this, is why does the equilibrium favor the carbonyl, even though you lose aromaticity? Remember that aromaticity is an extremely stabilizing effect. Aromatic rings are more stable. So my question to you is, why is it favored when we lose aromaticity? And I'll give you a moment to pause the video and see if you can figure out why. And the reason is that in the enol form, we of course have an enol. We have an enol, and enols are terribly unstable. They're extremely high energy, and in the video on in the video where we form imidazolone propionate through urokinase hydrolase, I used this example, and I'll use it again. Let's say we have acetone, and acetone is actually something that we make through ketone body biosynthesis, right? And the equilibrium is going to greatly favor the ketone form, right? But on some level, you're going to have the enol form. And it turns out that the enol form is probably only about 0.01% of the total amount in terms of the species. You're going to have much more ketone, right? You're going to have much more, in other words, carbonyl, right? You're going to have much more of the carbonyl, and only about 0.01% of the enol, okay? So enols are terribly unstable, okay? And it turns out that there's more of an instability in the enol form than there is in the non-aromatic form. So in other words, it's more stable to be without aromaticity than it is to be with the enol. So the tautomerization will cause the enol to form into a carbonyl. And that's pretty handy because the very next enzyme is a serine hydrolase. Now let me ask you this. Let's say for instance that we are trying to go from here to here, right? So in other words, we're trying to go from this imidazolone propionate enol to this structure right here. And this is N-formimidawyl glutamate. Okay, this is N-formimidawyl glutamate. And my question to you is, how would we get from the enol imidazolone propionate to N-formimidawyl glutamate? And the problem is you can't do it directly, right? The reaction, which is imidazolone propionase, so this reaction right here, this one is imidazolone propionase, make that sure that's a P. Imidazolone propionase is a serine hydrolase. And what do you know about serine hydrolases? And certainly go back and watch the video on that if you need a refresher. But serine hydrolases require a carbonyl. They require carbonyl. They're going to do a nucleophilic acyl substitution reaction, and you can't do a nucleophilic acyl substitution reaction on an enol. So it's pretty handy that the equilibrium favors the carbonyl, even though we lose aromaticity. And the reason it's handy is because the imidazolone propionase is going to do the nucleophilic acyl substitution using the serine hydrolase mechanism. Okay. And once we generate N-formamidyl glutamate, you can see that there's a multitude of pathways that we can do, but what I want to do is focus on the one that's done most often in humans, and that's going to be this one right here. Okay. Now the enzyme that this is, let me write it down, it's called glutamate formamino, I'll put TAse for transferase. So it's glutamate formaminotransferase, okay? And it's a tetrahydrofolate dependent enzyme, so we can write tetrahydrofolate, right? It's a tetrahydrofolate dependent enzyme. And what you're going to get out of it is you're going to get N-formamino tetrahydrofolate, okay? So you're going to get N-formamino tetrahydrofolate. And we're not going to stop there because in reality there's not a whole lot we can do with N-formamidyl tetrahydrofolate, or we can call it N-formamino tetrahydrofolate. So we're going to have to convert it to other forms of tetrahydrofolate. And the enzyme that's going to consume N-formamino tetrahydrofolate is called Cyclo Deaminase. It's called Cyclo Deaminase, and I have a picture right down here. And Cyclo Deaminase is this enzyme right here. This is Cyclo Deaminase, and so what we're going to get out of it is called N5N10-methynyl tetrahydrofolate. Now, what I want to be perfectly clear about is that this is not N5N10-methylene tetrahydrofolate. That molecule is this one right here. Okay, what we have is N5N10. Let me write it. Methynyl, not methylene, methynyl tetrahydrofolate. Okay, we're going to have to convert it to something else. N5N10 methynyl tetrahydrofolate is not going to do us a whole lot of good. Okay, so what we're going to have to do is we're going to have to convert it in one of two ways. Okay, so we're going to take this. Let me write this. It's going to be N5N10 methynyl and then tetrahydrofolate. So we can take this one of two directions. Either we can react it with Cyclo Hydrolase or N5N10 methylene tetrahydrofolate dehydrogenase. So let's talk about the first one. Let's react it with Cyclo Hydrolase. Cyclo Hydrolase, and that's this enzyme right here. That's this enzyme. And what this reaction is essentially going to be is a Schiff-based hydrolysis. So this reaction, let me do it in light blue. This is a Schiff-based hydrolysis. Okay, and what you're going to get out of it is N10 formal tetrahydrofolate. Okay, and that can be used by several enzymes. Okay, and also N10 formal tetrahydrofolate is also a product of N10 formal tetrahydrofolate synthetase. Okay, so there's multiple ways to get this form of tetrahydrofolate. Okay, and I might also mention this, that whenever you use N10 formal tetrahydrofolate, what you're essentially transferring is going to be an aldehyde. So you're going to transfer an aldehyde when you use this reaction, or if you use a reaction that transfers something from N10 formal tetrahydrofolate. Okay, so the other fate of N5N10 methynyl tetrahydrofolate is going to be reacting with N5N10 methylene, so this reaction is named for the reverse reaction, methylene tetrahydrofolate dehydrogenase. And if we're running it in the direction that goes to N5N10 methylene tetrahydrofolate, okay, we're going to have to consume an NADPH. Okay, so we're going to have to consume an NADPH. But that overall is going to be worth it, right? Because N5N10 methylene tetrahydrofolate is used not only in glycine catabolism, but it's also used in thymidylate synthase. And we've mentioned that enzyme here and there, and we'll see it again when we do nucleic acid biosynthesis. But thymidylate synthase is basically going to take deoxy-UMP and convert it to TMP. And then you can phosphorylate TMP, 2 times to get thymidine triphosphate, and that can be used in DNA replication. So it has important aspects other places. So not only does it have its place in glycine catabolism, but it's also involved in nucleic acid biosynthesis. Okay, so basically when we degrade histidine, okay, so let's come back up here. When we degrade histidine, we're not really getting anything in the form of like ATP or anything like that, okay? Notice that when we did histidine ammonia lyase, all we got was ammonia, right? When we did urocanate hydratase, we really didn't get anything net from that, right? Except for, imidazolone propionate. Imidazolone propionase really didn't give us anything, right? So the main end product of histidine catabolism is going to be, number one, glutamate, and we didn't mention that so far. So this is glutamate, right? So we get a glutamate, but the main energy yielding product is going to be some forms of tetrahydrofolate, and the immediate product of histidine catabolism is going to be this N-form amino tetrahydrofolate, but realize that molecule, which is shown right here, can be consumed by Cyclo Deaminase to form N5-N10 methynyl tetrahydrofolate. And then in general, that can react with N5-N10 methylene tetrahydrofolate dehydrogenase, which, it burns in NADPH, but we get an N5-N10 methylene tetrahydrofolate, and then that can be used in glycine catabolism, and then it also can be used in thymidylate synthesis, okay, through thymidylate synthase. So let's do one more quick recap. So we start with histidine, and then we essentially deaminate it to form an E configuration alkene, and that's characteristic of urocanate. Then we hydrate it, and we lose that alkene, and we end up with something called imidazolone propionate. And that is in the enol form, at least at the end of the mechanism of the enzyme. And it quickly tautomerizes to the non-aromatic carbonyl form. And that's going to react with imidazolone propionase, and that's going to give us N-formidinol glutamate. And then that's going to react with glutamate form aminotransferase, and so we get glutamate, but our main energy-yielding product is going to be N-form amino tetrahydrofolate. And go back and watch the video on tetrahydrofolate metabolism for a little bit more detail on this. Okay, so I hope this video gave you a little bit of intuition on histidine catabolism, and also realize histidine is not biosynthesized in humans, so that means that it is an essential amino acid. See you in the next video.